When the Human Genome Project completed its mapping of human DNA in 2003, scientists expected to find roughly 100,000 protein-coding genes, enough to explain the complexity of a human being. What they found was approximately 20,000, about the same number as a roundworm. More surprising was the functional accounting. Only 1.5 to 2% of the human genome codes for proteins. The remaining 98% had no identified function. Scientists working from a materialist model in which genetic information is purely chemical instruction code labeled it junk DNA, evolutionary debris, broken genes, genomic noise accumulated over millions of years. But here is where the story shifts. In 2012, the Decode project, a 10-year, 32-country, 442-scientist research collaboration, published its findings in Nature. The conclusion was categorical. At least 80% of the human genome is biochemically active. The 98% that was called junk is not junk. It is functional. It regulates when and how genes are expressed. It encodes structural information for RNA. It coordinates the timing of development. It responds dynamically to environmental signals. Calling it junk was not a scientific conclusion. It was a statement of the limits of the science at the time. What these 98% of DNA strands are actually doing is only beginning to be understood. And the early findings point toward a function that has no clean place in the materialist framework. They appear to be receiving and processing information from outside the cell. The next episode is already in my profile. Subscribe so you don't miss it. The work of molecular biologist Bruce Lipton, published across 3 decades and culminating in his 2005 book, The Biology of Belief, demonstrated through cell membrane research that the environment controls gene expression more powerfully than the genes themselves. The cell membrane contains protein receptors that act as antennas. They read the electromagnetic environment surrounding the cell and translate that information into gene activation or suppression signals. This is the mechanism of epigenetics. The same DNA sequence can produce radically different physical outcomes depending on the signals received from outside the cell. What signals? Electromagnetic fields, chemical gradients, temperature, and, critically, the bioelectrical field generated by the organism's own nervous system, which is itself shaped by consciousness and emotional state. HeartMath Institute researcher Roland McCready has measured DNA conformational changes in response to the electromagnetic fields generated by coherent versus incoherent emotional states. When a human being enters a state of genuine gratitude, the heart's electromagnetic field reorganizes in ways that measurably alter DNA winding state in nearby tissue. Russian biophysicist Pyotr Garjajev went further. His team demonstrated that DNA responds to coherent laser light and modulated radio frequencies in ways that appear to involve language-like information transfer. In multiple experiments, they showed that DNA could be reprogrammed by exposing it to frequency patterns that carried informational structure. Not chemical mutation, frequency-mediated reprogramming. Garjajev concluded that DNA functions as a biological receiver for frequencies far beyond the visible light spectrum, including frequencies that are, in principle, cosmic in origin. The next episode is already in my profile. Subscribe so you don't miss it. The cosmic ray connection is where this becomes impossible to dismiss. Galactic cosmic rays are high-energy charged particles originating from supernova explosions, pulsar winds, and the galactic center. They penetrate Earth's magnetic field continuously, with intensity variations corresponding to solar activity cycles and galactic positioning. Every living cell on Earth is continuously bathed in this radiation. Research published in the journal Astrobiology has documented measurable correlations between cosmic ray flux intensity and rates of genetic mutation in bacteria. Higher cosmic ray exposure produces higher rates of novel genetic variation, not random damage, but directed variation that increases biological diversity and adaptability. The implication is that the galactic environment actively participates in the evolution of life on Earth. The so-called junk DNA, the 98%, may be the receiver array for this galactic signal. Tightly coiled, mostly silent, waiting for the right frequency input to activate sequences that have been carried dormant in the genome for generations. Every 25,920 years, Earth passes through a different sector of the galaxy. The photon density, the cosmic ray flux, the quality of incoming galactic radiation changes. The ancient Maya, the Vedic astronomers, and the authors of the Egyptian Duat texts all describe this as a time of transformation, of the sleeping divine nature of humanity awakening. Not metaphor, not prophecy. Biology responding to a precisely timed cosmic signal, activating sequences in the human genome that have been waiting for exactly this frequency. And once you see this, you cannot unsee it. The next episode is already in my profile. Subscribe so you don't miss it.